Thank you so much, sir. Heading towards our session 4, which is the last session of the day. Yeah. I would like to invite our co-chairperson, Dr. Usha Johari, on the dais. Dr. Keruvi Lahiri on the dais. Moderator, Dr. Mamta Muranjan. Convener, Dr. Shruti Bajaj. And co-convener, Dr. Nipa Thakkar.
So I think we'll begin the first session. The first speaker in the current session is Professor Martin Jager from Netherlands, and he'll be speaking on the eye color and she'll be speaking on eye color and eye melanoma. Over to you, Professor Martin.
Ladies and gentlemen, it is a great pleasure to speak to you today and about the subject about eye color and uveal melanoma. I have no financial interest in this subject, and I very much thank Drs. Prakash Natarajan, Chola and Lahiri for inviting me to give this keynote address in the session Women in Eye Genetics Research at the first international conference of the Global Eye Genetics Consortium. I'm speaking to you from the Netherlands. These are all different people that have contributed to the research of my laboratory, and I'll be discussing their work. The disease I'm talking about is uveal melanoma, a disease like cutaneous melanoma deriving from pigment cells, but these pigment cells are in the eye, in the choroid. And it is a deadly disease. It's a malignant tumor, mainly in adults, in contrast with retinoblastoma, more known, but especially in children. Uveal melanoma has a low incidence, but a very high mortality rate, up to 50%, by spreading from the eye to the liver, giving rise to liver metastases. There is a wide range of risk factors. Quite a number of them have to deal with genetics, although we do not know exactly what the genes are. An elder age is a risk factor, also the presence of pigmented lesion around the eye, nevus of Ota, and Northern European ancestry, blonde hair, tendency to sunburn, lack inability to tan, a high number of freckles or a high number of moles, and a previous history of cutaneous melanoma. Another one is light eye color, as reviewed by one of my students, Dr. Houtzager. Uveal melanoma contain a small number of recurrent mutations, somatic mutations in the tumors themselves. Already all choroidal nevi and almost all melanomas have a mutation in two genes, GNAQ and GNA11. When it then develops into a uveal melanoma, there are also mutations in three major genes, BEP1, IFX1, SF3B1, and there are chromosome aberrations. The most malignant type comes from tumors with a BEP1 mutation. The gene on BEP1 is on chromosome 3, and those tumors lack one chromosome 3 and have a mutation in BEP1, ending in having no expression of messenger RNA or protein of the BEP1 gene. Now, this is about genetics, and a visible type of genetics is eye color. There are a real large number of eye colors, especially of the mixed forms, but one can really separate them going from blue to green brown to dark brown. Here is one eye color chart with 24 different eye colors, and we tried to divide our patients into different groups. Why were we interested in that? Well, it is a genetically determined factor, and there are at least a dozen different genes, but the most important ones are the HERC2 and the OCA2 locus. Other loci are shown here, and the genetics determine whether an eye has a specific pheomelanin or eumelanin. Pheomelanin is associated with blue eyes and green eyes and is more likely to develop damage to the DNA. There are three main categories, as I said. We divide it into blue-gray, intermediate, and brown eyes. And blue eyes are known to have the highest risk of developing uveal melanoma. Now, why is it easy for us to study this? Here is a map of the world with different locations and where one can find brown eyes and where blue eyes. Blue is especially found in Europe. Here's the percentage of people with blue eyes in Europe. And the lighter the color, the more people have blue eyes. Northern Europe has especially blue eyes, and there's an arrow pointing to the Netherlands. In the Netherlands, we have more than 50% of the people having blue eyes. I'm working in Leiden, and we could compare our group of patients with uveal melanoma with a large group of patients that were looked at in Rotterdam. And among our Leiden patients, it is especially striking that only 19% had green or hazel eyes, while in the population that is 8%, and brown eyes in our tumor population was 16%, and in the general population 23%. So we have fewer brown eyes and more blue, gray, and green eyes in our population. This has similarly been published of groups looking at people in Canada, Australia, France, Germany. In all the different countries, it was found that among the population of blue-eyed people, there are more uveal melanoma. There's a higher risk to develop uveal melanoma. Now, there are several risk factors for developing metastases. Well, here also, to develop metastases, patients have elder age, but also larger tumor size. Tumors that grow outside of the eye have a specific cell type and specific chromosome aberrations, monosomy 3 or extra copies of chromosome 8q. For monosomy 3, it is the BAP1 gene. For chromosome 8q, we do not know which gene is the important one there. One of my students, Mr. Dogrusos, reviewed this in Acta Ophthalmologica. Now, tumor size is a clear prognostic factor. You see three curves here. The top line, smallest tumors, the middle line, the medium-sized, and the lower line is the large tumors and the follow-up. The larger the tumor, the more chance of developing metastases and dying. Other prognostic factors are chromosome aberrations. I already mentioned chromosome 3 and the RNA expression pattern that comes out of this, as described several years ago by Onken and Harbour. They are known as class 1 and class 2. So when you look at this chromosome 3 status, here are two curves from Leiden with long-term follow-up. If one chromosome 3 is missing, the green line, monosomy, you have 80% of people dying, and when there is no monosomy, it's between 15 and 25% long-term. Now, based on many analyses, there are 4 different groups for prognosis, A, B, C, D, and A and B still have their chromosome 3 in order, disomy 3, and B has more copies of chromosome 8q. C and D both have BAP1 loss and loss of chromosome 3. Group D, in addition, has a lot of inflammation. A and B are known as class 1, C and D are known as class 2 and are associated with bad prognosis. Now, in a beautiful paper, Dr. Carol Shields in the Indian Journal of Ophthalmology analyzed those 4 groups. And the 4 groups are here going, if you look at liver metastases development, blue is A, reddish brown is B, green is group C, and the lowest group is group D. So these genetic groups all defined the amount of the chance of patients developing metastases and subsequently dying from them. And the higher the lettering, the worse the prognosis. We now wondered, looking at eye color, whether that influenced prognosis. We had a database with over 400 patients where we knew both the histopathology, the chromosome status, and the eye color. 65% of the patients were blue, 19% green, and 15% brown. So we looked at, is eye color related to survival? And then also, does it have anything to do with chromosome aberrations? Here you see the 3 curves of survival. And although brown is a little bit lower than the other 2, blue stands for blue eyes, green for green eyes, these differences were not significant between the eye colors. So people with different eye colors had a similar prognosis. But then we looked deeper. We looked at the effect of the chromosome 3 BAP1 loss in the tumors of the people with the different eye colors. And now if you look at the middle curve, those are the people with green or hazel eyes. If they had a normal chromosome 3 status, hardly anybody developed metastases. While you see on the left and the right that there were higher numbers of people developing metastases. But the bigger difference is with the chromosome 3 status. If a tumor in a person with green eyes developed chromosome 3 loss, BAP1 loss, the chance of dying from that tumor became very, very high. So the difference in those eyes between not having chromosome 3 loss and having chromosome 3 loss was about 75% chance of dying. On the left, the gray eyes, you see there is a significant difference, but when you look on the right, you see the brown eyes, and there was no significant difference in survival depending on the in relation to the chromosome 3 status. And here we see the different groups as already described by Dr. Shields, the A, B, C, and D category. And these are the data from Shields, a large database of more than 1,000 cases. For blue eyes, you can see that the expected categorization and survival is really obvious, but for brown eyes, A and B do not really differ, and C and D do not really differ. So what has been written in many papers over the years coming from Northern Europe and Northern America was true for blue-eyed people, but it was not so clear for the brown-eyed individuals. All these were Caucasians. Now, this makes me come to the conclusion. There have been several papers in Asia about uveal melanoma in Asia. The group of Dr. Ketan wrote a nice review about Indian perspective, and they wrote that despite that UM is a rarity in Asia, it is imperative for an ophthalmologist to recognize the clinical entity. And that is because also in Asia it can be a deadly disease. Together with Dr. Shields, Dr. Manshukoda and Dr. Singh wrote a review about the incidence. The incidence of uveal melanoma in Asia is much lower than in Europe, but it does occur, as I already mentioned. But the patients are younger. Mean age at presentation in Asia is 40 to 55 years, while in Europe it's often 58 years. studies have shown an increased risk for metastases with epithelioid tumors and inflammatory phenotype, as has been beautiful. With that, I want to thank the LUMC Ocular Oncology team for having so many patients and allowing the lab to use the material for analyses. And thank you very much for your attention, and have a great Congress.
Thank you for that excellent talk. I would like to introduce the next speaker, Dr. Jessie Berry. She's an associate professor of ophthalmology and dermatology, the division chief in ocular oncology services. She has received her undergraduate and medical degree from Harvard University. She's going to today speak about liquid biopsy application for retinoblastoma, from diagnosis to prognosis of ocular salvage. Thank you.
Children's Hospital Los Angeles, and the University of Southern California. I'd like to thank the organizers for inviting me today to speak on retinoblastoma liquid biopsy applications, from diagnosis to prognosis of ocular salvage. These are my financial disclosures. We all know that unlike most tumors, retinoblastoma is not directly biopsied, and that's due to concern for extraocular tumor seeding. However, a lack of biopsy does have real-world clinical impact. There are no eye-specific diagnostic or prognostic biomarkers. We use absolutely no personalized treatment medicine for retinoblastoma, and there's a real limited understanding of in vivo tumorigenesis, or how these tumors change at a molecular level under therapeutic pressure. To overcome this, we took the opportunity that was afforded to us by the initiation of intravitreal injections, at which point aqueous humor is taken as a safety mechanism to lower the overall pressure of the eye. And using this safety enhanced mechanism, there have been no documented cases of extraocular tumor spread. And in 2017, our laboratory was the first to state that the aqueous humor could be used as a surrogate to tumor biopsy, or as a liquid biopsy for retinoblastoma. And subsequent to that, we've been able to answer some really important clinical questions in this application. The first is there DNA in the aqueous? The answer is clearly yes. This has been demonstrated by our lab, as well as validated by other laboratories around the world. We've standardized now our specimen handling and collection methodology. We've shown that greater than 80% of the DNA in the aqueous is tumor-derived. And in fact, about 95% of aqueous humor samples have tumor DNA at some concentration. And in fact, if you take the aqueous humor at diagnosis, when there's a larger tumor burden, you can look for tumor DNA two ways, both via detection of the RB1 gene as well as chromosomal alterations, and you get 100% yield. Next question, can the aqueous play a role in diagnosis? Yes. So from that tumor DNA, you can of course sequence the RB1 mutation. From the aqueous humor as a liquid biopsy, there is enough DNA to determine both if there are two somatic mutations or the presence of loss of heterozygosity with high fidelity from a single sample. And in fact, we've shown that you can use this not only to identify the RB1 mutations, but also the chromosomal alterations from a single sample. This, of course, leads us to lots of other clinical questions that we face as people who treat patients with retinoblastoma, like what do you do when there's loss of view, in the diagnostic dilemmas that, while rare, still do exist, and can we use this to guide further therapy? Those questions are still ongoing, but we think the answer is yes. Can the aqueous play a role in prognosis for saving the eye? This is the most important question to me, and we think the answer so far seems to be yes. What really bothered me as a clinician, and other people have said the same thing, is that two children will come in and they look exactly alike clinically. They're treated more or less with the same treatment regimen because, again, we don't personalize our treatment schemes. And one patient will do really well, the eye will be salvaged, and the other will persist and recur until they finally require a secondary enucleation to prevent tumor spread. But can we predict this better now that we have access to molecular biomarkers? So we analyzed this directly in 2 papers. The most recent was our follow-up paper on over 50 eyes with over 116 aqueous humor samples. And we were able to compare 2 groups, the patients who saved their eyes with treatment and the patients who required enucleation. And what we found by comparing those 2 groups, which previously was not possible because you only had tumor DNA when the eye was enucleated, we were able to show that gain of chromosome 6p was not only more commonly found in eyes that required enucleation from treatment failure, but the amplitude or the number of copies of that chromosome was higher there. And in fact, as a clinician, I wanted to know how much that matters. So if you do traditional staging, A, B, C, D, E, or AJCC staging, you can see that Group D eyes in green and Group E eyes in yellow, of course, have lower salvage rates, about 62 and 33%. And this was using either intraarterial or systemic chemotherapy. However, if instead of prognosticating them clinically, you used molecular prognostication based on 6p, you can see that only 9% of eyes with 6p in the aqueous humor were able to be salvaged. And in fact, there was a 10 times increased odd of that eye failing therapy and requiring secondary enucleation. That to me is a very impactful prognostic test. Can the aqueous play a role in monitoring for therapeutic response in the eye? The answer is yes. Right now, we all know that we monitor this pretty much subjectively, looking at the tumor burden and the response to the clinical treatment in the eye. But we can use the aqueous as a molecular objective test. And we've shown that by monitoring tumor fraction, or the percentage of tumor DNA that persists in the aqueous humor, we can show that the eye is responding to therapy as the tumor fraction falls or stays high. As the tumor fraction does not move, the tumor is not responding. Furthermore, a fall and then an increase in tumor fraction portends an intraocular recurrence before it can be seen by the clinician. Does blood play a role in liquid biopsy for RB? The answer here again is yes. There have been several centers around the world looking at this, but some of the most recent and exciting publications are from Sloan Kettering. They looked at an advanced group of eyes at diagnosis and at varying times throughout therapy, and they are looking for the presence of RB1 mutant DNA, or those somatic mutations in the blood. They have found it at a relatively low tumor fraction, as would be expected in the blood. But the real question that we need to answer for blood is, what does a negative blood test mean? About 50% of these children at diagnosis had 0 mutant RB1 DNA in the blood, and about 60% were negative during active therapy. So how do we apply that going forward clinically? That's something that we all need to join together as a community to decide. Can you just use the blood? In my humble opinion, the answer here is no. And the reason for that is twofold. First of all, we published in Ophthalmology that aqueous is superior to the blood as a liquid biopsy for RB. That doesn't mean that the blood can't play a role, but rather what we found is that the tumor fraction, or the percent of tumor DNA, is significantly higher when you look at matched blood and aqueous humor samples. And not only is it higher, remember that 40% of patients have bilateral disease. So in those patients, it's important to be able to specify biomarkers per eye, and that can only be done with the aqueous. This is an example of a patient with bilateral disease, different prognostic biomarkers in each eye, none of which were found in the blood because there just wasn't a high enough tumor fraction. And you can see there's the 6p gain that we suggested was prognostic, and this is the eye that failed therapy, while this eye did not. We're also able to find aqueous tumor-derived DNA in very low tumor burden eyes, including Group B. Finally, are there other eye-specific biomarkers in the aqueous humor? The answer is yes. This has been a whole new and exciting realm of research for us, including microRNA and extracellular vesicles, which I'm not going to get into, but just know that aqueous has all sorts of things in there that are really exciting in terms of retinoblastoma, care delivery, and personalized medicine. There was a recent review by Jarish et al., the impact of cell-free DNA analysis on the management of retinoblastoma. If you're interested in it, I think it was a beautiful review of the status of liquid biopsy for RB. I don't do this alone. I have to thank my wonderful retinoblastoma biopsy lab team, including, of course, our grant funding sources. Thank you so much. Hello, I'm Jess Berry from Children's Hospital Los Angeles, and
So thank you everyone for the patient listening. That was talked by Dr. Jessie Berry from USA. Now we have Dr. Shilpa Bisht. She's online. Oh, she has joined us online, and I would take this opportunity to briefly introduce her. She's a postdoctoral fellow from Johns Hopkins Medicine, PhD in the Department of Anatomy from All India Institute of Medical Sciences, MSc Molecular and Human Genetics from the Department of Human and Molecular Genetics, BSc Technology, HNB Garhwal University. So with that introduction, over to her for the online presentation.
Hello everyone,
Got it.
my name is Dr. Shilpa Bisht and I am currently working as a postdoctoral fellow at the Department of Oncology, Johns Hopkins Medicine. It gives me immense pleasure to present my work today at the first International Congress of Global Eye Genetics Consortium. The title of my today's talk is The Role of Targeted Exome Sequencing in Retinoblastoma, and this work was carried out under the esteemed supervision of Dr. Bhavna Chawla, Department of Ophthalmology, AIIMS New Delhi, and Dr. Rima Dada, Department of Anatomy, AIIMS New Delhi. So I would like to start with a brief introduction about retinoblastoma. So retinoblastoma is the most common childhood intraocular malignancy, and it is first proposed to be caused by Knudsen's two-hit hypothesis, which was given in the year 1971 by Alfred G. Knudsen. And according to this hypothesis, the loss of function mutation in both the alleles of RB1 tumor suppressor gene is the main cause of RB1 tumors. So now coming to the RB statistics, the worldwide incidence of RB is about 1 case in every 15,000 to 18,000 live births, which corresponds to about approximately 3% of all the solid tumors and approximately 2.5 to 4% of all the pediatric malignancies. In India, every year 1,400 to 1,800 new RB cases were diagnosed every year. And according to a recent trend, there is a 30% increase in the RB incidence over the past 5 years. Now coming to the RB epidemiology. So in this map, you can see that the highest incidence of RB is found to be in regions with low socioeconomic status, such as Southeast Asia and South America, where about 40 to 70% of RB patients die in Asia and Africa as compared to regions of higher socioeconomic status, such as Europe, Canada, and the US, where the mortality due to retinoblastoma is 3 to 5%, which is very less as compared to the regions with low socioeconomic status, such as Asia and Africa. Now coming to the classification of RB tumors. So RB tumors were generally classified as the familial tumors and the non-familial ones. So in familial RB tumors, which comprises about 40% of all the cases, in these patients, the mutation is inherited from an affected parent, which comprises most of the bilateral cases as well as 15% of all the unilateral RB cases. So the non-familial RB cases, which comprises 60% of all the RB cases, were further classified into sporadic heritable and sporadic non-heritable. So in the sporadic heritable tumors, the new germline mutation in the RB1 gene, it could occur by chance or as a result of pre-zygotic exposure of the either parent. So these tumors manifest themselves either as bilateral or the unilateral RB tumors. Whereas the sporadic non-heritable tumors are always unilateral RB tumors, and these are postulated to be caused by post-zygotic factors, which can lead to both somatic events in the RB1 tumor suppressor genes, which ultimately results in these unilateral RB tumors. So now coming to the function of the RB1 gene, which is the main gene causing the RB tumors, and also the protein encoded by the RB1 gene, which is known as the RB protein or the PRB. So the RB1 gene is basically a tumor suppressor gene with its cytogenetic location at 13 to 14.2 region. And it has been seen that 6 to 8% of all the RB cases shows chromosomal abnormality. The RB1 gene is quite a long gene with about 27 exons, and it encodes for RB protein or the PRB, which is a major G1 checkpoint regulator because it regulates the transition of the cell from the G1 to S phase of the cell cycle. So actually, RB binds with a family of E2F transcription factors, which are important regulators, and they are actually regulating the transcription of genes which are required for cell proliferation and also the transition of the cell from G1 to S phase of the cell cycle. So now coming to the initial signs and symptoms of retinoblastoma. So the most common sign of retinoblastoma occurrence is leukoporia or unusually pupillary appearance, as you see in these patients' pictures. Some cases also show squint nystagmus, change in visual status or loss of vision in few of the cases, with pain and swelling of the eyelids, protrusion of the eye, and glaucoma has also been seen in few of the RB patients. So the clinical features or the clinical manifestations between RB patients, it depends on the size of the deletion in the RB1 gene, which is located on the 13 to 14.2 chromosomal location. So now coming to the molecular genetic testing in the RB tumors. So RB actually represents a heterogeneous group of tumors at cellular, molecular, as well as genetic levels. And RB1 gene harbors a variety of mutations, and till now more than 928 mutations have been reported in the RB1 gene so far. And RB1 gene actually shows a variable mutation detection rate. And when you see the distribution of germline mutation across the RB1 gene, these are primarily nonsense mutations in nature, and these nonsense mutations are primarily aggregated in few subset of exons in the RB1 gene, which are known as the clusters or hotspots of mutations in the RB1 gene. So, as I already mentioned, that RB1 gene causation hypothesis was first of all proposed by the Knudsen's two-hit hypothesis in the year 1971, and according to this hypothesis, both the alleles of RB1 tumor suppressor gene were found to be mutated, which ultimately leads to the initiation of these tumors. But it was only until 2013 hypothesis came out from the Brenda L. Gelly group, which shows that it's not necessary that all the RB tumors are caused by loss of function mutation in the RB1 tumor suppressor gene, but there is a subset of unilateral RB tumors which comprises 2% of all the RB tumors, and these tumors are basically characterized by a different genomic and gene expression profile as compared to the classical RB1 tumors, and they do not have any mutations in the RB1 gene. They do have a normal RB expression, normal RB protein expression, and they are actually characterized by a very high number, a very high copy number of the MYC and oncogene. So MYC and oncogene is generally found to be having 28 or 30 copies in classical RB1 tumors, which are having biallelic mutation in the RB1 gene, whereas these tumors, which are specifically characterized by the Brenda L. Gelly group in year 2013, these are characterized by a very high copy number of the MYC and oncogene, and these tumors are having 121 to 130 copies of the MYC and oncogene, and these are having a very bad high-risk histopathological risk features, very poor aggressive retinoblastoma tumors, and they are having very early mean age onset as compared to the classical unilateral RB tumors. So this actually, this study actually challenged the central dogma which was proposed by the Knudsen two-hit hypothesis, that it's not necessary that always loss of function mutation in RB1 initiates the retinoblastoma tumors. That in addition to that, that there could be some other oncogenes, tumor suppressor genes, or cell cycle checkpoints, cell cycle regulatory genes that might have important role in the initiation and poor prognosis of these tumors. So what does MYC and oncogene do? It is actually a very important oncogene, and it is a very important regulator in the P53 MDM2 P14 ARF pathway, and regulates the transition of the cell from G1 to S phase of the cell cycle. So now for the current study, we did the mutation analysis. So we did the polymerase chain reaction and the Sanger sequencing, and also the DNA sequencing. We also did a very important next generation sequencing technology, that is a targeted exome sequencing technology using the Illumina MiSeq technology. And these are basically the steps of the targeted exome sequencing that we employed for the current study, which includes the basic steps, which are preparation of sample, preparation of the DNA libraries, and selection and purification of the hybridized targets, and then quality check of the target regions, resequencing, and then finally the validation by the PCR Sanger sequencing technology. So for the current study, we actually recruited the retinoblastoma patients according to certain inclusion and exclusion criteria. The inclusion criteria for RB patient includes Only the children which are diagnosed with unilateral RB intraocular tumors between the age of 0 to 5 years have been included. RB patients with no history of chemotherapy, radiotherapy, or intraarterial melphalan, and these patients does not reported with any other diseases or any major surgery. The exclusion criteria for the RB patients is patients with extraocular disease, metastatic RB, or any cases with a positive family history of RB were completely excluded from the study. The inclusion criteria for healthy children which serve as controls includes healthy children with no history of any systemic illness, any history of cancer, or any other ocular diseases were recruited as the controls. So now these are the clinical pathological parameters of the RB patients that have been included in the study. So we recruited 75 sporadic unilateral RB patients after obtaining ethical clearance from the AIMS Institutional Ethics Committee. And as I already mentioned that about 6 to 8% of the retinoblastoma patients showed cytogenetic abnormality. So with this, we actually did the karyotypic analysis for both numerical as well as structural abnormalities in the 13q14.2 chromosomal region, which actually harbors the RB1 tumors of Resser gene. But we did not found any cytogenetic abnormality in like any of the patients that have been analyzed. And these are the representative G-bended metaphase spread from RB patient male and female, which shows that it has a completely normal chromosomal 13q14.2 chromosomal region that harbors the RB1 gene. So with this, we further analyzed the patient samples using the PCR Sanger sequencing and targeted exome sequencing technologies. And as I already mentioned that there are few RB1 mutational hotspots where the frequency of RB1 mutation occurrence is higher as compared to the other regions. So on the basis of previous studies, we actually selected 4 mutational hotspots in the RB1 gene. We did not sequence the whole RB1 gene because it is quite heterogeneous and it might have some intronic regions or intronic variations which could not be detected due to PCR Sanger sequencing. So we actually chose only 4 mutation hotspots in the RB1 gene. And after PCR Sanger sequencing, we found 2 novel mutations in the RB1 gene, out of which one is a frame shift mutation and the other one is a variant. And none of the controls had these mutations that we found in the RB patients. It actually expands our current knowledge regarding the genomic landscape of RB tumors, and it also highlights that there is a necessity of doing the high resolution techniques such as next generation sequencing technology, such as targeted exome sequencing for the detection of novel variants that could have like important role in the pathogenesis of retinoblastoma tumors, because next generation sequencing technologies have a very high resolution as compared to the conventional PCR Sanger sequencing technologies, and they might help to determine new variants or new polymorphisms that could have important role in poor prognosis and very early mean age of onset in unilateral RB tumors. So with this, I will end my presentation here, and with the interest of time, I will stop here. I would like to thank the committee members for inviting me to present my PhD thesis work here. Thank you very much, and thanks a lot for your patience. Thank you.
We go on to the next speaker. We have Dr. Kerry E. Goads from USA, who will be talking on casting a wide net to engage participants for rare disease registries. She is completed her Masters of Science in Genetics and Developmental Biology, and she is also Bachelor of Multidisciplinary Studies in Professional Writing, Business and Communications, Bachelor of Science in Animal and Veterinary Science, and currently she is an Associate Director, Office of Data Science and Health Informatics. Over to you, Dr. Kerry.
Institutes of Health, and I'm going to be presenting today about casting a wide net for engaging participants with rare diseases in registries. Much of the research that happens at the National Eye Institute involves trying to move research from the bench to the bedside, or how can we make scientific discovery benefit patients? In particular, in the branch that I'm in, we look to figure out how to deliver on the promise of genetics to stimulate more discoveries. And particularly in the projects that I've worked for, we want to know how we can empower people with their genetic results and connect them with research. For my presentation, I'm going to give some information about the iGene project at the NEI, which is a US and Canadian biobank, patient registry, and clinical data resource for the vision health community. I'm also going to talk a little bit about data infrastructure and data sharing, and then wrap up the talk with what I see as some opportunities for international collaborations. So many of you might already be familiar with the iGene program, which is a network resource that consists of a repository of DNA and blood samples, a registry for patient contact information and storing consents, including different opt-in, opt-out options, and then a database for research use, including phenotype data and genotype data. iGene began in September of 2006, recruiting participants, and ended in December of 2015. And over the course of that time span, we recruited 6,403 participants with over 30 different inherited eye conditions. And that's depicted in this graph that shows our peak of 2011, followed by a short drop, and again, increasing in 2015. iGene used a unique study design where we allowed sites throughout the US and Canada to either decide to do their own IRB approved protocol or rely on the NEI protocol to do consenting of their patients that they identified as eligible for the study. And this was really beneficial to the participants. You can see here that we had participants recruited from almost all of the states in the United States, and a lot of the recruitment sites actually correspond to places where there weren't a lot of ophthalmologists or optometrists. And this is important to consider in rare disease research because some ophthalmologists or optometrists, some eye care providers have never seen some of these conditions in their entire life. And so by providing them a low bar to identifying participants that could be eligible and benefit from being part of the network was really crucial to the success of iGene. So despite having a large area of ability to participate and a low barrier to entry for clinicians to help get their patients connected to the network, patients still traveled really long distances to get involved in the iGene network. And you can see here in the chart on the left, it shows the residence of the participant, so actually where they lived, connected to the green dots, which are where they were enrolled in the network. And on average of our 6,400 plus participants, the average travel was 400 kilometers. And you can see by the red lines, the denser the red lines, the more people traveled that far. And you can see actually on the map of the United States, some people were going from Florida to California, or even, traveling from Alaska to Michigan to be engaged in the project. So for all of the participants in iGene, the referring clinician would enter some clinical information about their exam, which would be reviewed by our clinical review staff in the iGene Coordinating Center. We would take a blood sample that was submitted to our central coordinating lab, create DNA samples for biobanking, and then send a portion of that sample off to CLIA labs, which are the US equivalent of the certification needed for submitting clinical test results back to participants. So for iGene, we have tested around 96% of participants. Most of those have been in clinical labs. but also many have been sent for research use. The people who have not been tested yet, there's usually a reason for that at this point. Maybe they don't quite match a criteria for phenotype of something that would be tested in one of our network labs. Or it could be that they're the relative of a participant that had genetic testing that was inconclusive. That's also an option. So this table just shows some solved rates, and this is granted from a couple years ago, but most of the time for retinitis pigmentosa and other retinal degenerations, we're solving those cases around a little over 50% of the time. And if you look through this chart, you'll see things like, pattern dystrophy, which have a much lower rate of being quote unquote solved, and things like retinoschisis, which is typically a one gene associated condition, have a higher rate of solved. So this goes to show that even though we have completed genetic testing on most of the participants in iGene, there's still a lot of research questions to be asked. And I'll talk a little bit about the process we have for allowing research access to this information. So anyone who has a vision-related research question relevant to the data that's available in iGene is welcome to submit an application for research use. Applications require just a brief summary of the intention of the study and the affiliations of the applicant and how they will share data back to iGene. So this is a crucial component of the project where individuals who are using the data agree to provide information back. So we're continuing to add knowledge to the database. So far, we've had 32 applications for use. 14 of those requested DNA access, and 10 actually requested the iGene Coordinating Center contact participants. And we've allowed the participants to opt in or opt out of being contacted for additional research studies for which they might be eligible for. And this has been really helpful because for recruiting for rare diseases can be almost like looking for a needle in a haystack. And we've created this large capture network to allow connection to participants and engage them in the research process. So this has been a very successful component of the iGene network, the ability to connect back with participants. And just another quick note about that, the contact back to participants is done completely through our coordinating centers. So the participant then has the autonomy to decide whether or not to act on that information that we have provided them. We never give out contact information directly to third parties. Scientific areas of our secondary research use have obviously been in gene discovery, genotype-phenotype correlation. Some have created iPSC lines from individuals involved in the project. Some image analysis, AI, deep learning, disease predictions, typical things like that. And then looking at genetic modifiers, enrolling for natural history studies, and that kind of thing. And there's one other comment about the research use is that our review committee that looks at the whether or not to approve or disapprove a project is instructed to just look at whether or not the data available in the resource in the iGene network is sufficient to address the project, but not rating the project on scientific feasibility or anything like that. Essentially, if there's a hypothesis that can be tested with iGene and no, ethical concerns about the usage and it complies with the participant's consent, then that should be an approved request. And some accomplishments of creating the network is many, many publications, but more importantly, we've been able to connect with participants, over 200 participants for other studies. Participants actually receive the results of the genetic testing. They receive a certified clinical genetic testing report and genetic counseling as an agreement with the enrolling providers, which is a huge benefit to the participants. We've also identified through research results, pathogenic or likely pathogenic variants in over 200 participants. So being part of the research project also has its benefits for potential future research level results that we can then verify in a clinical lab to submit back to the patients. So recently we took a look at the currently registered clinical trials that are involving inherited eye conditions for genes that we tested, and we actually quantified how many participants could potentially benefit from additional trials based on knowing their results that we had submitted back to them through their participation on iGene. And this table just shows a short snapshot of that. It's included in a recent paper we wrote in the supplemental section. But we calculated that at least 1,600 participants could be eligible for other trials just based on knowing the results of what we provided back to them. And we looked at this at variant specific row level. Now I'm just going to talk a little bit about the infrastructure we used, which is an important component of any research project that aims to share broadly. So we use a application called BRICS, or the Biomedical Research Informatics Computing Software. And this is an NIH supported software that houses several of our studies now at the National Eye Institute. So recently we have added APIs to the platform, and we've done this because as our data gets bigger, we now have millions of rows of genetic information. It becomes cumbersome for people to log in and use the application for a lot of data science projects using a graphical user interface. And so we wanted to be able to more easily connect with scripting softwares such as, R, which is now known as POSIT, and Jupyter notebooks, and Python and things like that. So by using APIs, we're allowed to authenticate folks in a scripting manner and have them pull the information from the backend server into whatever workflow that they're using. And this has been a benefit to our more technologically advanced computer science savvy community. However, we still need the user interface for a lot of other users who are just trying to find quick stats, and filter and join different forms in the system, or join even different research studies. We've also added row level access controls, and this is to address participant consent options. So, some people have agreed to contribute their information for broad research use versus vision health only research use. And by allowing these row level access options for our data management staff, this allows us to honor those patient participant level requests for their use of their data. So now I'll shift gears a little bit more and talk about potential opportunities for international collaboration. And one thing that we've been involved with in the NEI Office of Data Science and Health Informatics is improving the use of standards and interoperability for not only research and biomedical research use, clinical trials, but also in the electronic health record space and data collected on US patients who are visiting a clinical practice. And so some of those are improving standard use of LOINC and SNOMED, and improving ocular data formatting for FHIR and image DICOM standardization are all areas that the NEI has been focusing on that will hopefully allow more interoperability between international partners. And at the end is my contact. So if you're interested in discussing those further, I'm happy to connect. So on a more granular level, the things that I think are the low-hanging fruit for international collaborations that can extend the net for rare disease research and make kind of the vision of the iGene project and engaging participants more fruitful across the globe are collaborations and coordination around data collection protocols, standard operating procedures, international collaboration for creating common data dictionaries and representations of typical eye exams, including the data elements and specific measurements of, how to collect visual acuity and interocular pressure and those kinds of things. What are exactly the parameters of those measures? And leveraging existing standards instead of creating project by project data elements to increase interoperability. And by doing this, we would be able to develop a worldwide network of clinical centers and clinical trials and clinical data sharing that wouldn't require data to leave any countries. Row level data could stay where it is, and we wouldn't need shared platforms or any of that. Just interoperability kind of solves the issue of centralized resources. And so with that, I'll thank you for your time. This QR code links to my Linktree. It has my contact information if you're interested in discussing anything further. And thanks again for the invitation.
Dr. Bhavna, who's the chairperson of this session, she wanted a minute to address all of you all. Usko jara online kar dena. Bhavna, are you there?
Yeah, can you hear me?
Yeah, we can hear you.
Okay, excellent. Thank you so much. I mean, really, it's such a pleasure and a privilege to be here, here in spirit, though, because physically I was not able to make it. But congratulations to all the organizers for a wonderful meeting. I would have loved to be here in person. So actually, I'm also born and brought up in Bombay, and I never miss an opportunity to miss an invitation from Mumbai. Unfortunately, due to certain last minute circumstances, I couldn't make it here. But congratulations to all of you for the wonderful show. And I've been hearing and enjoying all the presentations. And I'm really especially thankful to Dr. John Prakash and to Dr. Natarajan in particular, because when they asked me to put together a session, the idea that came to my mind is like, women doing so much of wonderful work all over the world in every field. Why not we have a separate session for women in, eye genetics and research? And all of you have really lived up to it with all these wonderful presentations. I mean, it couldn't have been better. And special thanks to Dr. Karobi for getting us all together, putting up this whole session and this wonderful show. I hope you enjoy the rest of the meeting. And next time, for sure, I'll do my best to be in person. Thank you all.
Bhavna, thank you so much.
Bhavna, no excuse, no?
No, I didn't know that you were listening.
I'm listening, I'm watching.
Okay.
And Bhavna, you can not only be with us in spirit, you can down yourself in spirits also.
Oh, great. That's
So
even more
where did you grow
wonderful.
up in Bombay?
Matunga, very close to your center.
Where in Matunga?
So, next to Don Bosco High School,
Okay, that's where the hospital is.
VJTI
Yes.
and city. So I lived in the campus.
Oh, I know VGT.
Yeah. Yeah.
I
So
love that
used
campus.
so I used to speak better Marathi than Hindi, but it's only after coming to Delhi that, I had to switch my loyalties.
Oh, but you can come back to Bombay.
Of course, anytime.
And we have our genetic lab in Khalsa College in the National Facility for Biotechnology. I think there are some students here in Khalsa College also. And we have there are two researchers are sitting there. You should mentor them, Shruti and Samruddhi.
Yeah, I was
They're
just
on the
listening
stage.
to all the wonderful talks. I'm completely glued to this program, not going anywhere.
Thank you, Bhavna.
Thank you.
And also you can tell about the new society which we are forming where Dr. Lady is a pediatric ophthalmologist. She deals with that.
Yeah, definitely. As we get a little bit more organized, we're able to formalize it. We'll definitely involve everyone, and we need everybody.
You can tell the name now.
Okay, so we are going to launch very soon the Eye Cancer Society of India. Basically, I've been working in the area of ocular oncology and retinoblastoma for many years, and I'm seeking Professor Natarajan's support to help us, in this mission and this endeavor. And we will all be reaching out to you very soon with the details of the society, how everyone can join and contribute. And we would love to have everyone on board. Thank you.
Thank you, Professor Chawla. Hope to see you soon in Bombay.
Thank you.
I would like to introduce our next speaker, Dr. Sunita Birjania. Dr. Sunita Birjania is a genetic consultant based in New Delhi who offers counseling to patients or relatives at risk of inherited disorders and advice of the consequences and nature of the disorder. She offers her expertise on both diagnostic and supportive aspects. Dr. Sunita is a part of the Center of Medical Genetics at the Sir Gangaram Hospital and follows dedication to her patients, giving them the care and attention they require. Dr. Sunita Birjania.
Thank you for the kind introduction. At the outset, I would like to thank all the organizers, Professor Natarajan, Dr. Lahiri, and everybody who has given me this opportunity. May I seek permission to share my slides?
Yes, please. Go ahead.
Okay, thank you. Okay. So good evening, everyone. So the topic that has been assigned to me is congenital cataracts. And as per the previous speakers and also Dr. Bajaj's, congenital cataract seems to be a really important cause of, pediatric illness in ophthalmology. So cataracts, as we all know, are opacifications of the lens, and pediatric cataracts are common. Nearly 10% cases of childhood blindness worldwide is attributed to cataracts. Congenital is the word when we use when the lens opacity is present in the first year of life. 1 in 250 newborns have some congenital cataract, though they may not be clinically significant. There are various causes, genetic and non-genetic, which we will be discussing about. And the number of genes have been progressively increasing. As we can see in the CatMap website, there are more than 100 genes now which are associated with cataracts. So congenital cataracts can be divided into zonular, polar, total, and membranes. I need not elaborate this in front of this august audience who are ophthalmologists. So I'll just go ahead. So among the non-genetic etiology, the large group of maternal infections, such as the torch group of infections, the toxoplasmosis, rubella, CMV, and also the others like syphilis, Coxsackie, herpes simplex, they're all implicated in causing cataracts, as well as maternal drug intake like corticosteroids, and very rarely now thalidomide may produce cataracts. There are instances of cataracts happening on maternal radiation exposure. Cataracts can also happen due to trauma or other conditions of the eye, such as uveitis. So amongst the genetic causes which perform the large majority of the etiology in pediatrics, like 25 to 50% are hereditary, 70% are non-syndromic congenital cataracts, 15% of these cataracts have some other ophthalmologic findings, which I will be telling, and 15% have multisystemic. So these are the genes that are involved. There are a few of the genes that are involved in congenital cataracts. The list keeps on going on and on. These are the crystalline related genes in this. Just moving ahead. I'm not able to move my slide. Okay.
I know it's not moving.
Yeah, so I just moved to the other one. So these are some more of the genes. So now coming to basically these cataracts may not occur alone. So they can be combined with other problems in the eye, such as the anterior segment dysgenesis, microphthalmia, the ectopia, even aniridia. So I'll just put forth one case, which is quite interesting. This was a young couple. I'll just show. Yeah. So this was a young couple who came presented to us for pre-conception genetic counseling in view of the fact that both of them were visibly blind. So the husband was a 31-year-old male with vision loss and nystagmus noted since infancy. An ERG performed at AIMS, New Delhi at 3 years of age had given him the diagnosis of Leber's congenital amaurosis. Nevertheless, the parents guided him further, and with a good education, he's actually a lecturer at Delhi University. So he did have bilateral cataracts when he was 28 years old, and he got operated, following which he is still getting treated for glaucoma. Now, he is the one, and his own sister also has a similar illness. And two of his paternal cousins also have similar illness. Coming to the wife, she was also blind, but her blindness was due to bilateral cataracts that were discovered at birth in both the eyes, and which she got operated in childhood. Then there was no associated history of any hearing loss or any other malformations or any suspicion of any metabolic disorder like galactosemia. There was no developmental delay. She actually attended a normal school, and she could read and write well until she was 20 years old, when she developed glaucoma, for which she required surgery. And currently there is perception of light in the right and some finger counting in the left. So in her family also, there was a strong history of congenital cataracts. Both her sisters, her father, her uncle, and the uncle's son had congenital cataracts. So this was a huge family history of congenital cataracts, which was showing a pattern of autosomal dominant inheritance. So genetic testing was performed in the couple, both the husband and wife, to determine a genetic cause. So the husband was noted to have pathogenic homozygous variant in the RPGRIP1 gene, which causes Leber congenital amaurosis type 6. So in the wife, the NGS panel detected a heterozygous likely disease-causing variant in the exon 6 of the CRY-BB1 gene. This was a 7 base pair deletion, which causes a frame shift and ultimately terminates the protein much earlier than expected. So mutations in this gene are associated with CTR-CT17 type of congenital zonular central nuclear opacities, along with microphthalmia, glaucoma, amblyopia, and strabismus, quite similar to what the patient had. So as we all know, the lens transparency is maintained because of many proteins, including the crystallins, connexins, the MIP and LIM2, crystallins being alpha, beta, and gamma. There are various genes in the crystalline responsible. And here we are with our own patient having a variant, a heterozygous variant in the CRY-BB1 gene. So thus, it is expected that her family members who are affected would be having the same mutation. So the implications are that because they had sought genetic counseling, coming to the husband's condition, this was an autosomal recessive condition, which means that both the copies of his gene of RPGRIP1 were defective. So we know that the wife was tested and she does not carry any problem in this gene. So in their offsprings, all the children will receive at least one affected copy, but they will also receive one healthy copy from the wife. And thus, because not both of the copies are affected, all the children are going to be fine. So here there is no transmission of disease, and there is no need for any prenatal diagnosis when it comes to children or the pregnancy. Now, when we come to this wife's condition, which was an autosomal dominant condition, the condition is brought about by the affection of only one of the two copies. So that's the dominant part. So here the husband does not have any defect in any of his genes. So when there is pregnancy, the children are going to receive either the defective gene or the healthy gene. So it is a 50-50 chance. So there is a 50% risk of passing on congenital cataract due to the defective copy of the gene alone from the wife. So we counseled the couple likewise, and the couple accepted the risk, and they went ahead with the pregnancy, and they did not opt for any prenatal diagnosis, knowing that this was a condition which was treatable. So that's how they went around. So I'm not sure what happened to the child, but our counseling perhaps worked. So the cataracts can be associated with other multisystemic disorders as well. Most prominently are the ones which, inborn errors of metabolism and many of the syndromic ones, as we have been seeing all along in the day. So they can carry any pattern of inheritance. We have already seen the two in the last case, and they could be X-linked as well. So lots of disorders are there which are associated with cataracts alone. And metabolic group is very important because this is the only group that has very effective treatment. So that's why galactosemia comes right on top. And I will not be elucidating others. As you can see, there are multi-systems like from muscle, CNS, connective tissue, renal, dermatological, any other. Many of the chromosomal disorders, like Dr. Shruti's talk mentioned children with Down syndrome presenting with cataract. So this is how it is. We must not miss these cases and not just concentrate on cataract. So lots of IEMs do feature cataract as one of the signs, and starting with galactosemia, Lowy syndrome, peroxisomal disorders, then other disorders. So I'll just illustrate two cases very briefly. This was a 9-month-old boy referred to us from Jaipur, born to non-consanguineous healthy couple, but from the same community. So this boy presented with bilateral cataract when he was about 2 months old, and he got operated at 3 and 4 months of age. And he had aphakia, and that's all. But in addition, the parents noted that he was not really doing very well. At 9 months, he did not have a proper neck control, and he was only sitting with support. He was otherwise quite alert. He was vocalizing, etc. The other thing that was worrisome for the parents was his poor weight gain. He had had no seizures, no vomiting, no acute illnesses as such. So in the family, interestingly, there was another genetic disorder in the husband's brother, and that was cystic megalencephaly. So they were really worried about this boy developing the uncle's disease, but we had to tell them that this was not likely because his head size was in fact on the lower side and his MRI had been normal. So upon investigation, because of the cataracts and the global delay, urine was first tested, which was positive for the non-glucose reducing substance, and the galactose in the blood was also raised. So we did the enzyme assays first for the GAL1P transferase and then galactokinase. So galactokinase deficiency was observed, and the genetic testing confirmed the diagnosis of galactokinase deficiency. So this boy was then treated with lactose-galactose-free diet, that is basically avoidance of milk and milk products, and with regular therapies for enhancement of his development. Now he's 6 years old and he's doing quite well on a milk-restricted, galactose-restricted diet. So the second child was born to consanguineous, this was a consanguineous family, and they had come to us with their fourth child, having lost 2 children earlier. The first child actually is healthy. The second girl had died in the neonatal period because of perinatal asphyxia. The third boy developed cataracts at birth. He also had developmental delay, and he died at 18 months of age because of some prolonged respiratory illness. So no further investigations were available for this boy. Now this boy, when we saw, he presented with at 5 months of age, and he already had bilateral cataracts. He did have developmental delay and also failure to thrive. So cutting the long story short, we investigated him further. The routine investigations, the blood parameters, CBC, liver and kidney functions were all normal. Metabolic investigations also were pretty much normal, except that his lactate was very high. It was repeated. So there was a suspicion of, because it's a multisystemic disorder with failure to thrive, even cataracts, and also developmental delay, a mitochondrial disorder was high on our mind. And this is a multi-gene disorder. If any of the 300, 400 nuclear genes could be involved in this case. So a next generation sequencing was performed, and 2 mutations were noted in the AGK gene. And after doing the test on the parents, that is called segregation. So one parent was harboring one variant each, and that thus confirming the biallelic affection in the child. So the child was ultimately diagnosed to have Sanger syndrome, and which features with congenital cataracts, hypertrophic cardiomyopathy, which went unnoticed in our child, muscle weakness, and lactic acidosis. So both of these conditions actually say that we have to look further than the eye whenever there are cataracts. So just a little diagnostic approach to patients with congenital cataracts we've taken from our textbook of genetics. So starting again from history of any trauma, radiation exposure, any systemic disease, whether maternal or child exposure to drugs. Very important to take family history of cataract. So there we have in case 1 extensive family history. Eye examination, as we all know, is a must for such patients, and as also the evaluation by pediatrician and the geneticist. So a whole host of laboratory tests can be done for evaluation, but one could, of course, streamline all the investigations. And ultimately, it's the genetic test that would give you the diagnosis. So the protocol for genetic testing is also basically goes with pre-test counseling and confirming the suspicion of the disease before going ahead with the genetic testing. Like we say, good phenotyping is a must. And then genetic testing is nothing but a blood test, but of course it involves time and money. And not all cases do get confirmed by this. The diagnostic yield varies from anywhere from 50 to 70%, like let's say. So just to take home a message, genetic testing must be offered in all genetic eye diseases such as cataracts, because they are useful in understanding the natural history. Once you have a confirmed diagnosis, you know what it is. You can tell the parents what you expect in the future. If it is treatable, you can start treatment and you can prognosticate them. Then systemic care in genetic eye diseases becomes important. Then you can, of course, provide genetic counseling for recurrence risks, etc. One could screen all the family members who are at risk for such a condition, especially the dominant ones. And of course, carrier testing, if positive, then can lead to prenatal diagnosis. And that's the only way of prevention. And of course, we have lots of hope for the future. So thus research is important. So with that, I end my talk, and thank you very much for the opportunity.
Thank you, Dr. Sunita. That was an excellent talk and a good overview of congenital cataracts. And it was a welcome relief to hear about cataracts after all the dry degenerations and myopias that we've been hearing throughout the day. So thank you so much. And another special thank you to you and Mamta, because Mamta gave me your reference and you said yes at the first go without a fuss or a bother. So I'm very thankful to you. And it was an excellent talk, Sunita.
Thank you, ma'am. Ma'am, may I just mention, I would like to apologize on behalf of Dr. Ratnapuri, who couldn't make it. She's not feeling well, so probably she missed out. Really sorry.
Yeah, she spoke to me today morning,
Yeah.
and I asked her whether she would like to give it tomorrow at some time. So she said she would let me know.
Yeah, thank you so much.
Thank you so much. Convey my regards to her and ask her to get well soon.
Thank you, ma'am.
And I thank all the delegates who are there here. You all have been patiently listening throughout the day, and hats off to all your patients. I call this meeting to a close today, and we come back refreshed and rejuvenated for the meeting tomorrow. Thank you so much. Yes. Mamta, will you give? Of course. This is for Dr. Usha Johari.
Thank you, madam, for coming.
Bhavna will receive it virtually. And this, yeah. Yeah, give her this bouquet also. And who else was to be given? So give all these lovely ladies one. Okay,
Yeah.
just give her also. She can take it and give it to her brother at home. And
Come for a quick
Usha,
photograph.
Mamta, yeah.
Madam, all of it.
Yes. This is a nice. Yeah,
Yeah.
sweet all, yeah.
Yeah.
I'm towering above you, Mati.
Yes, thank you.
Towering inferno.
Yeah.